Hello, my name is Bernie Schwartz, Commercial Manager in the Water and Lab Products team within Thermo Fisher Scientific. I've been helping customers outfit their labs with the most up-to-date and reliable lab essentials for over 30 years. Today, I would like to talk to you about every lab, everyday benchtop products that play an essential role in the SARS-CoV-2 sample preparation workflow. We are going to consult with some knowledgeable colleagues to answer some common questions. Every lab, everyday products are used across the SARS-CoV-2 workflow, including vaccine development and research. Some of the needs include mixing and homogenization of samples for RT-PCR and ELISA tests for tubes and 96 or 384 well plates, gentle mixing of cultured cells to grow the virus, removal of solvents from samples such as oligos used in diagnostic test kits, and maintaining a constant temperature. Let's take a closer look at some of this equipment and further the discussion. The Vortex mixer is a common piece of equipment in all labs. Precise mixing to help ensure homogeny is an important step of sample preparation. The Thermo Scientific Vortex mixer offers digital speed and time controls, allowing the user to program the Vortex mixer to precise time and RPM specified in protocols, continuous and touch modes for various protocols, and multiple tube holder accessories for processing various vial types and quantities. Mr. Fosket, is there a recommended time and speed to Vortex a SARS-CoV-2 sample?
There is no real set speed or time. As with all experiments, it is key to have consistency across the testing protocol, making sure all reagents are mixed completely.
Would this be the same as sample prep for real-time PCR?
Yes, it is really important to homogenize the tissue sample completely in preparation for the amplification stage of the experiment.
The microplate shaker is used for mixing samples at high speed for high throughput screening of small molecules. An example of this would be screening for the ELISA protocols. The Thermo Scientific microplate shaker offers digital speed and time controls which allow users to program the microplate shaker to precise time and RPM specified in protocols. The standard platform fits 4 microplates of SPD and ANSI standard footprint design, and the optional large assembly platform that fits up to 6 microplates. Mr. Fosket, is the Thermo Scientific microplate shaker suitable in the screening process?
Yes, definitely. The use of a microplate shaker is an important workhorse in the laboratory. The need for agitation to mix reagents to a constant, reliable state. The design of the unit to enable small volumes to be mixed comes from the design of the mechanism to obtain the correct throw.
With 96 well or 384 well microplate standard in RT-PCR and ELISA tests, why is homogenization important with SARS-CoV-2?
With mass testing and the use of liquid handling robots with small volume samples, it's very important to mix the samples precisely and across all wells.
SARS-CoV-2 samples are enveloped positive sense single-stranded RNA viruses. In the production of diagnostic test kits, a key component will be an oligo or synthetic strand of DNA RNA to make up a primer. These oligos are often contained in ammonia hydroxide, a solution of water and ammonia, and need to be dried down. Other solvents can also be used here, such as methanol, water, and ethanol. A vacuum concentrator, or speed vac, is used in sample preparation for solvent removal. The Thermo Scientific SPD 2030 offers a preheat button that allows user to preheat the chamber to 45°C, a lid lock indicator for product integrity, digital displays for heat settings, run heat time, and vacuum pressure, preset programs by solvent type, and user-defined programs, giving the end user 6 program options. Mr. Sochi, what else can you tell us about the capabilities of the SPD 2030?
Hi, Bernie. Yeah, that's a very good question. The SPD 2030 is a very versatile evaporation system. First off, it's a fully integrated system. We kind of call it the works in a box. It incorporates a speed vac chamber, an oil-free vacuum pump, and a minus 50-degree cold trap. It's what we also refer to as a plug-and-play system, meaning simply you unbox the system, place it on a standard benchtop, It has a very small footprint, takes up less than 3 feet. You plug it in, you turn it on, and you're ready to go.
What other applications is it suited to?
It's another very good question, Bernie. The SPD 2030 is well suited for all types of biological samples that one may need to dry down before they are analyzed. Samples such as proteins, enzymes, DNA, RNA, HPLC fractions, as well as many other types of living cells or plant material.
Can acids be used with the SPD 2030?
This is a very good question, Bernie. I get this all the time. And the simple answer is yes. But it has to be in very small percentages. Percentages such as 0.01% and maybe slightly higher. And of course, they need to be diluted with other solvents. Solvents such as methanol, acetonitrile, ethanol, even water. That's the only way you could be able to use acids in an SPD 2030.
An important aspect of testing is maintaining constant temperature for RNA reverse transcription and DNA amplification. An example would be ELISA at a specific temperature. The Thermo Scientific portfolio of dry baths offer a wide range of temperature options to support your unique needs. For repeatability, there are easy-to-use programs to support multiple users. Startup options include either immediate start after the timer is set or after the desired temperature is reached to help the viability of your sample. There is also added versatility with easy-to-change blocks. Ideal for DNA extractions, enzymatic reactions, or reactions that need to be processed at or near room temperature, as well as reagent warming or cooling. Mr. Foscat, why is it important for a heat block to maintain a constant temperature for RNA reverse transcription and DNA amplification?
Amplification occurs at a set temperature. Dry baths need to be constructed so the block will give an even temperature and the controller pushes the heat to the correct temperature set point slowly without over temp or under temp deviations so not to affect the experiment.
How does that affect SARS-CoV-2 samples?
Dry baths are used within the SARS workflow, once to heat the reagents to gain the correct reaction, and secondly they can be used for heat inactivation of the virus to enable safe handling of the sample for further manipulation.
I hope this has demonstrated how every lab, everyday benchtop products can play an essential role in the SARS-CoV-2 sample preparation workflow. Thank you for your time.